I just want to quickly introduce the CTSA leadership representative at this table. Brian Currie is Vice President for Medical Research at Montefiore, which has also invested 10 years of hard-earned marginal dollars on building a remarkable infrastructure in the EMR, and not ready to talk about all of its aspects, but certainly that's one of the important steps in our CTSA building blocks. Dan Stein runs our human biorepository and analytical lab. Dan does diabetes. Brian's an infectious disease nut. Another infectious disease person, Marla Keller, runs our CRC on the East Campus. She's in the Department of Medicine and is a microbicide expert. And finally, Sanjeev Gupta, who's our connection to the world of stem cell research and novel methodologies, and he's a liver expert. So we have a diversity of backgrounds up here, and probably a diversity of backgrounds and opinions and questions in the audience. So I think what I'll ask you all to do is to perhaps begin the questions, starting at that end with Sanjeev. For either, I could probably consume the entire 45 minutes that we have with questions for these two speakers, but why don't you go ahead and get us started?
So you're giving me 45 minutes?
I'm giving you two minutes to state your case.
First, I enjoyed both the talks. They were absolutely marvelous and gave a wonderful opening to raise a number of questions, of course. And first, Dr. Mitchell, Ms. Mitchell, I'm assuming, raised the issues of, I think, private-private partnerships, because here we were talking about entrepreneurship rather than what is an alternative model of public-private partnerships as well, of course, where governments set up certain relationships. And from a healthcare scenario point of view, we know that there are certain simple things that have more profound effects than certain complex technologies. And I'm not necessarily less than any other person using technology, but we know that behavioral modifications, obesity, guinea worm, this, that, malaria. So from those concepts coming from the academia, where if I'm a typical representative of many of these people, and if we want to be high-minded, we say, we create ideas and we want to create solutions that can be implemented. And again, taking the example of genetic sequencing, if it costs $1,000 only per person, we are looking at $300 billion for 300 million American population. In the current healthcare crisis, that's not really doable. So how do you think, in a traditional academic scenario, the combination of these ideas and the intrinsic tension within them can be solved?
That's a simple question.
That's a way complicated question. Unfortunately, as you heard from Dan, we're a long way from personalized medicine. And I think we're going to get there through rare diseases and pediatrics. But my friends in Big Pharma think we're going to get there a different road. But frankly, we just have to do these things. I had a question, Dan, that I think ties back to this. Okay. So are you working with the folks in Utah that have built?
I'm familiar with what they've built. There are a number of other places around the country that are trying to do this. Duke has a program. Each program has big pluses and big holes. Yeah. And there are sort of vague and early stage alliances being created to sort of trying to figure out how to do this at a national level. I will say that in the short term, this is there's no way that this saves money, but in the long term, you have to sort of figure in how much it costs per adverse drug reaction, for example. There is data that says that adverse drug reactions are the 4th to 6th leading cause of death in hospitalized patients in this country, 100,000 deaths a year. So if you could put a dent in those, if you could reduce that by 50%, through these kinds of approaches, and when I say these kinds of approaches, I mean information science as well as genomic science. So a lot of those deaths are not due to genetically determined adverse drug reactions. They're due to the wrong drug for the wrong patient at the wrong time and the wrong dose. That's the antithesis of personalized medicine. So a nursing error or a physician error or a handwriting error or a system error. So information science has the capability of dealing with that part of it as well. So I think at the end of the day, it's very important to rope in the economics guys, because the models that you have to use to try to figure out at what point this becomes cost effective or not are interesting. But it costs a couple $100 to do a UGT1A1 or a TPMT or a CYP2D6 or a CYP2C9 right now. So if you can, for the same amount of money, you can get lots more sequence information. It seems like that would be the way to do it, although how to store that, how to access it, how much it costs to do all those things have to be figured into that as well.
And the cost of non-response that you alluded to.
Right. So let me go back to answer his question, because I was hoping Dan was going to say this. So to come back to your question, I mean, what you just heard from him in his very first responses is there's a lot of people doing things in a lot of different ways. And, rather than talk about healthcare reform, I would like to talk about NIH reform. And the ability to actually create these massive collaborations that will make a difference, instead of a whole bunch of little collaborations that are structured in different ways. And, as we've said in discussions at the NIH for years, the person who has the gold gets to make the rules. And we need to get there. Well, and if, in fact, we can do some of these major collaborations, whether they're public-private partnerships, university philanthropy partnerships, frankly, I don't care. They need to have protocols and standards, et cetera, behind them, of which we don't have a lot of. But until we get there, we're never going to get closer to lower-cost healthcare.
So if I may ask...
I'm going to take the chairman's prerogative and look for some audience participation here. Bill. That's Bill J. Identify yourselves. I'll agreement to the mic if you don't make yourselves heard.
Okay, so I'm Bill Jacobson. I'm a professor here in the micro department. I'm interested in the intellectual property question. It used to be that when I went to scientific meetings, people would tell new data. Recently, for the last 5 years, they don't tell anything unless it's published. And it goes back to this intellectual property thing. The initial snip that he showed that's waiting for intellectual property to be discovered was clearly present in those original patents, that were found. But yet you said they were worthless. So I want to know, when do you go from, how do we go forward in this intellectual property environment that is equitable and makes people want to collaborate?
No, I absolutely agree with you. And that was, I don't know if you were here when I was talking about my Bob Cook-Deegan story, or if you were here, relative to, I don't think we know today exactly when we should be patenting and when we shouldn't be patenting, which is why we need to get closer to figuring out what are these pre-competitive arenas where we need to share data much more widely. The unfortunate answer is I don't know the answer for you. I think we're getting closer to knowing the answer, which is why the people who get involved in helping you make these decisions about what you should and shouldn't patent need to be very, very, very experienced and entrenched in the state of the art of what needs to be patented and what doesn't. So I sympathize with your comment, and it absolutely is the reason that we started Science Commons and the reason that we're really pushing for these collaborations. And I think, frankly, at the end of the day, I'll come back to my previous comment about the people who have the gold rule. Part of what my friends in the philanthropy circles are doing is they're attaching rules to some of the philanthropy money that they're giving, relative to, I want to push this forward into a collaboration versus you going out and personally patenting your IP. So I think we're seeing lots of different kinds of experiences and experiments all over the place, which is not helpful except for to tell you, again, expertise you getting access to outside expertise is the most important thing that needs to happen when you have that question.
The informaticians are all in Bethesda, actually, as we speak, right? And NIH has actually convened a big conclave to discuss
That's where my guy is.
Yes, and our guys too. So we went from crushing eggshells to 128-bit encryption, and I hardly know what all of this means. Marla.
As a follow-up to Bill's question, my question for Lesa is... Sorry. My question for Lesa is the university industry collaboration project that you mentioned, which involves 85 universities, and I think you mentioned over 60 companies. Is there an example of a project where there's collaboration to streamline the process regarding IP or licensing? Can you give an example of...
Yeah, and I'm trying to think. The project was led out of Georgia Tech and was in the IT space. And essentially they created, what we ended up finding, I'll go back to my Rich Newton story, where the industry thinks we mean one thing about sponsored research and the university thinks we mean another. And so essentially they created a software package decision tree that was designed both by the industry people and by the university people to help you understand when you're even setting out on a discussion between an agreement between university and industry, you need to answer a whole, and have a lot of knowledge to even ask the right questions is the problem. And be able to answer those questions in a way to figure out, am I going down a road that's going to get me to IP or not? Am I going down a road that's industry-sponsored research and the IP should be here? So we're trying to get to more science around his point, but starting at a very high level of that university-industry relation. And they created something called a turbo negotiator, which is literally a software package that those guys are using in the IT space. And it's easier on the IT side.
Yeah, it is easier. There are groups that are naturally collaborative. IT is one of them. Pediatricians are the others.
Yes.
I'm looking at our team from the business and commercialization side, which are represented at our CTSA table, and I'm reminded that, in fact, NIH claims to have a model template now for industry partnership on IT-relevant science. But I think the question you raise is, in fact, the important one. Is NIH really the barrier or the enabler for the transition of ideas into that space?
And literally the reason for doing the turbo negotiator was that we found no one was using that agreement.
Right.
How about conflict of interest in the process? I asked her to comment on conflict of interest in the process she's describing.
We're not supposed to mention the C word.
That's commercialization, I think.
I know.
Yeah, that's right.
Well, we keep talking about...
I'll use Bill Brody's. There's no conflict, there's no interest.
to IP or not to IP. The idea is actually to get the product into the clinical environment if it is efficacious and it should be there. So somewhere along the trail of development, we're going to go from intellectual property and concept, proof of concept, into clinical trials and into development to bring it to completion. And when it's all in one entity, there's always a concern about managing conflict of interest. The people who develop the compound are in fact part of the consortium that's going to do the clinical trials, that will do the analysis and eventually determine whether we see the compound or not. Are part of these arrangements?
Oh, sure. I mean, there's a lot, I would assume. Vanderbilt, I know Stanford, Dr. Richard Popp, who does the conflict of interest stuff at Stanford. I mean, there's lots of best practices that have been defined in this space. And frankly, I think you have much less of an issue on the therapeutic side than my friends on the device side do. Where it's a lot scarier in conflict of interest. But I think there are plenty of best practices out there.
Well, there are plenty of them out there, but every time you pick up a newspaper, there's another victim falling to that conflict of interest lately. So it's a
I think it's more about our lawyers than it is about our doctors.
Yeah, you're probably right.
Or senators from the Midwest.
Yes, exactly.
Dr. Roth?
conflict of interest is a poor term. It
Yeah.
might be competing interests or multiplicity of interests.
Right.
May be a better way to describe it, because a conflict of interest by definition is a fight. And so I think multiplicity of interest is a better term and really describes a broader set of effects that all of us are guilty of having multiplicity of interests.
Right.
Some more than others,
And
apparently.
making those transparent would be the key. Yeah, that's right.
Hopefully. So, as
Yes.
staying on a theme, I'll get back to the questions. I'd like to sort of throw this a little bit more in Dan's perspective, though not necessarily experience. You know, Tom Friedman, since you read the New York media, I'm sorry I missed including the New Yorker. The world is flat. I do too. The world is flat, and so globalization has come. Academe has been bemoaning the lost opportunities in phase 2 and phase 3 that have gone abroad. So is science going to follow that? Are the interesting scientific studies that would be coupled with clinical trials, for example, genomics and pharmacogenomics, also going through the way of, or do we have to develop medical schools in Singapore that are affiliated with this?
No, no. There is a lot of interest, obviously, in pharmacogenomics worldwide. I think the United States is still ahead in a lot of aspects. The Europeans have a sense that they can work together, and they certainly have built some very, very large resources like the Wellcome Trust. But there is a collaboration that goes on between them and the United States. Japan is another area that has a big interest in this. The problem with the Japan Biobank is that it's not EMR based, so they have an army of ascertainers that go out into their 67 participating hospitals that cover 300,000 DNA samples and find cases and controls for when you want to do this. The reason I know about the Japan Biobank is that comes back to something that Lisa said, and that is that I've been part of the pharmacogenetics network since 2001, and we're now in a phase where they're recompeting the network. So I don't know if we'll be part of it. I hope we'll be part of it. But the NIH philosophy with regard to the PGRN has sort of evolved over that decade in an interesting way. The initial network was hardly a network. It was 10 different sites, each one of which did their own pharmacogenetic thing. And after about 5 years, we realized that that was probably the most effective way of advancing the field. So each center that was part of the network sort of committed. It was like a religious revival. Not that I've ever been to one of those, but committed to devoting some of their psychic energy to the betterment of the field. So we've created alliances. We view ourselves as the torchbearers for pharmacogenetics, and whatever alliance we can make with industry that hasn't worked very well. We tried industry with other people around the world who are doing this, with other groups within the United States that are doing it that are not formal parts of the pharmacogenetics network. We've encouraged that. So in the latest round of renewals, that came through in the RFA loud and clear, both in words as well as in dollars. We do pay attention to dollars. So the size of the grants was actually ratcheted down. And the NIH said, well, we're gonna keep some of the money for the network in reserve to fund network-wide projects or projects that the network wants to do with other sites. So the idea is that this is a mechanism that they have of sort of advancing the field. So I would argue that at least for the area of pharmacogenomics, the NIH has been very, very helpful in terms of pushing a particular agenda forward, and the kinds of agendas that people within the network have are the kinds of agendas I outline. It's not just a matter of discovering, your favorite SNP and your favorite drug, but actually understanding how it is we're going to develop CLIA-approved tests for this, how we're going to move this to the bedside, what the economic models are, what to pay attention to and what not to pay attention to. Those are the kinds of things. What are the ethical issues around this? And how to create international networks. We have a proposal in to create a network that will involve all the biobanks that are within the network, plus the Japan biobank, plus some biobanks in Europe that will do pharmacogenomics. So we hope that they're an enabler for this as opposed to a barrier. But
there were
we'll
some
see.
Questions. Ed?
Hi. Question for Dr. Roden. You mentioned the current funding you're having for your robot, for some of the SNP analysis, et cetera, which is all federal, very nice. But how much of a dollar investment did the hospital put in to set up the biobank, to set up the informatics, and the whole system?
Yeah, one of these years I'm going to be able to actually answer that question. So I've been asked that before, usually in a forum like this. My guess,
Executive Dean, be careful.
my guess is over the course of the last 5 years, it's probably been around $5 million. Something like that. Now, so the robot is federal. Some of the how do you cost account the informatics when there's a huge informatics infrastructure that drives our electronic medical record? They're there. I mean, they have to be there. So we piggyback onto them, and obviously we cost them something. And there's faculty time, and there's programmer time, and all that sort of thing. So it's a little hard to cost account, but it's something like that. And mostly people, as you would imagine, but the DNA extraction exercise, we do everything at Vanderbilt through institutional cores. So we don't have a separate core that holds the DNA samples. They're held in a DNA resource core that sort of, obviously we're the 900-pound gorilla in the corner for that resource, but that resource has about 100,000 other samples held for various investigators, and it's a really nice facility because they have ways of protecting the samples, duplicating the samples, barcoding the samples, so they know where everything is, and they won't release a sample without an IRB approval. Those kinds of safeguards. But when we started to accrue, we rapidly sort of exhausted their capability of doing DNA extraction. So we had to add another shift to the. We literally added an overnight shift, and we had to buy a second AutoPure extraction device for $250,000. So those are the kinds of institutional investments that.
So Harry Jacobs handshake.
Harry Jacobson, yeah. Oh yeah, no, it was I expect that he realized that he's an entrepreneur, so that's I don't think he thought this was going to come cheap, but at the end of the day, we think when we add up the V'ger stuff, this new stimulus grant that we got, the robot, it probably comes out as a wash, but it's not really a wash. I mean, the robot had to be there. It's not, but there are indirects associated with some of this as well. Not with the equipment.
Can I make another comment on that same topic? So I know where you're going, and I totally agree and understand. And when we decided to give our big grant to start the Institute for Advancing Medical Innovation of about $8.5 million, we went to the endowment of the university. And we said, we're ready to do this, and we've been working with you guys for a long time, but we're only doing this if you put in equal money. Because if the leadership of your university doesn't get this and isn't going to be supportive of doing this over time when it's going to be hard and we're going to have conflicts, et cetera, we don't want to do this. And it took a while. But and frankly, one of the proof of concept centers that I've been working with, the guys down in San Diego, have had a significant leadership issue. And the center down there is survived because of the community, because of leading scientists, entrepreneurs in the community who have come in to write some good checks because of the outcomes that we literally were able to show in our study. So I think it's so important that you have a commitment from the entity.
Let me just say one other thing that although there's been an investment, and that investment has to continue. There is a budget that I have every year for this particular project, and the fight is always exactly who's going to be funded off federal money and who's going to be funded institutionally so we can grow in different directions. It's not much of a fight because there is institutional support behind this. But one of the things that we obviously hope for is that this will be a springboard for a lot more federal dollars to come in through R01 mechanisms and other mechanisms. And I have a file in my computer of letters of support for BioView because I have a template, but I always sort of try to. I can always make it personal for the investigator because I can go to the record counter and say we have this many cases of this for you. And you'll need to work with the informatics guys to figure out who they are. But I've written 24 of those letters in the last year.
So you guys are getting the flavor that we need from you as a consultation here. So this is really terrific. And the questions we're asking, despite the dean's temporary absence, and he'll be back for the latter half of the program, is really that these are important issues for us at Montefiore and at Einstein as we build this infrastructure and we learn from people who've gone before us, and there are probably some opportunities that we've missed and don't want to sort of jump into a pool that's on its way to being emptied. Dan, actually there were a couple of other questions. Richard?
Yes, so my question is about integrating data into things like BioView that is not well ascertained by the health record. So there are some conditions like migraine or cognitive decline, some outcomes like calculated quality of life and symptoms, some really common conditions like depression, where the medical record ascertains them poorly. So if you solve the problem of linking patient-reported data to a biorepository.
Deeper phenotyping.
Deeper
right?
phenotyping. No, so the advantage and disadvantage of the model that I've presented to you is that what you see is what you get. What's in the medical record is what you get, and that's it. There's no. The disadvantage, of course, is that for Bill, there's no going back to the families. I mean, because we can't. We just don't do that. And as you know, maybe things are different here. I bet you they're not. The family history is not the family history is not a big part of the medical record. So what this project has done for us is highlight the deficiencies in our medical record in terms of using it as a research resource. And so you can imagine a system where you would engage people through the kinds of portals that I described, or when people check into the clinics now, they check in electronically. There are these little kiosks. And so you could actually imagine where people could actually enter new data into the electronic record, all of which eventually becomes part of BioView after it's de-identified. So we think that by highlighting the deficiencies in the electronic record in terms of how we describe the cases, we may be able to have a dent at that. At the end of the day, though, you're asking clinicians to do things that in an environment where they have real time constraints. So we think the right way to do it is probably engaging the patients. But the answer is yes.
On a similar note, we've seen the same thing. I'm working with Steven Spielberg with the Institute for Pediatric Innovation, and in the space of pediatric oncology, we know who every single doc is. And interestingly, my friends down the street are the founders of the company, of the electronic medical company that is in almost all pediatric hospitals in the United States. And so we've sat with them. Exactly. So we've sat with them and their design team, and they're working with the pediatric oncologists to literally think through, what do we have missing here that we need? And let's start doing some of this in our early stage trials and managing that.
I mean, we do case report forms all the time for new drug entities, and you could imagine where you could fill out the analogous thing every time somebody comes to clinic. There could be pull down lists, checklists. When I go to clinic, most of my medical record is template driven.
Really?
So there are ways to improve the electronic record.
Yes.
That should is.
As these biobanks start to grow, genotyping becomes more and more complicated. NIH has become interested in our ethical obligations as well as the practical issues of possibly contacting because of clinically significant genetic markers we found. And this is something that we often struggle with. So I was wondering about your experiences in terms of thinking things that you have set up for recontacting de-identified patient data and
So the de-identified model precludes recontact. I mean, that's the way it is. So when we started this, 5 years ago, the notion was, well, it's unlikely 5 years ago we would do a genetic test that we would ever want to recontact anybody with. And so it just shows how naive we were about understanding the pace at which this science has evolved. It's a big issue. We think that we may be lucky in the sense that by the time the dense genotyping becomes a reality, we'll have moved from the de-identified to the re-identified side. We're always asked, what do you do if you find something that tells? The example that people always use is BRCA1. What happens if you find somebody who has a BRCA1 mutation? Well, the answer is right now we have no way of recontacting them, and we say if we think that BRCA1 testing is clinically indicated, then that person should have BRCA1 testing on the re-identified side. It's sort of a cop-out, I understand, and it's the tip of the iceberg, because as resequencing comes online, as this exomic resequencing comes online, it's going to do it unbelievably quickly. Those kinds of issues are going to come to the fore. I was telling Bill yesterday that when I'm not thinking about this, I take care of patients with a disease or a set of diseases where the phenotype is a young healthy person who drops dead, and those diseases have a genetic basis. And the problem is that if I screen in a family, or if I screen across people who have no family history, I will find variants that might or might not indicate that they have the disease. Those variants are highly incompletely penetrant. So most people, for example, with congenital long QT syndrome, most mutation carriers never have a symptom in their life. So if you tell everybody who has a mutation, oh, by the way, you have this disease, you certainly haven't done anything for the healthcare bill, nor have you done anything for their mental well-being, unless, of course, you happen to pick up somebody who would have dropped dead. So it's going to be a very, very complicated set of issues. And I know I'm being parochial by talking about the long QT stuff, but I think the phenotype is so dramatic there. For BRCA1, you can tell somebody, get mammograms.
Sure, and that ties into what you can. The move towards prevention is all about identifying things.
But at the end of the day, we're not going to make anything cheaper by doing exomic resequencing. That's pretty sure, at least early on, until we figure out what all this means. So the issue. I mean, all I can say is that we have an ethics board that's external. We have internal ethicists who are play in the national space, and we worry about this problem a lot. Right now, we don't have genotyping data that would make us worry about it too much.
I'm going to take one more comment from Dan Stein, so we can break briefly.
Okay, this is
brief
really
comment and
directed to
hopefully
both Dan and Lesa. I think the emerging field of the GWAS studies, it's really coming to fruition, and perhaps that explains 10% of what we know in terms of disease. I was delighted to see your PheWAS approach, which I think really is the wave of the future. So I have a two-part related question. One is, we have all this phenotype, genotype data that's potentially out there with the hundreds of millions that pharmaceutical companies have spent. So for Lesa, what is actually occurring now to link that information with those of us in academia? Are they partnering? Are they releasing that data? It seems like that's a huge investment that they're just sitting on. And then the second part, and as someone involved in our biobank, it seems one has to be very careful in terms of targeting your resources in terms of very well annotated samples, and you're obviously doing in terms of a generalized patient record, but what are your criteria in terms of understanding if there's good phenotyping or not? Obviously you can't take every single piece of tissue and store it away.
And when is enough
And when is enough?
Okay,
and that's another good point.
Okay, so my answer is depressing. Hopefully his is good. And unfortunately, I mean, I totally understand the issue, and it's all about lawyers with the pharmaceutical industry. I mean, Lilly has really led the pack Genzyme has led the pack, but Lilly has led the pack as of late in opening up some of their compound libraries for, I think they have 4 university partners.
And CTSA is actually building that out.
Yeah, exactly. So I think we're starting. We will have hiccups because there will be findings in drugs that are already on the market that will cause front page news. And unfortunately, that's bad, but that is why they are as worried as they are, obviously. I mean, the good news / bad news is Big Pharma is falling apart. And it is really for everybody to understand that the Big Pharma model based upon blockbusters and lots of drugs and personalized medicine don't go together in terms of their structure. And so you could consider that potentially a good news, bad news thing, that as it falls apart and we're going to see different building blocks put back together again that might look more like our biological friends, it will become easier for them to collaborate. I think I am obviously just a wildly optimistic person in general, but I have some friends in the rare disease space that I think are really starting to thinking about banks. Jonathan Jacoby, I don't know if you know Jonathan, thinking about creating some rare disease banks, check in, check out, drugs, et cetera. And again, my point earlier in saying I have hope in rare diseases and pediatrics, there's no money in either one of those spaces. And so that's why I'm hoping we can go fast and low and nobody notices what we're doing. And we're going to make, hopefully some great successes, but it has to be in sharing and in this pre-competitive space. But again, Big Pharma they care, but they aren't that deep into this rare disease space. So I think it's going to be our space to prove the model, prove what's pre-competitive, what's not, and then go back to regular drugs.
I'm going to be brutally rude and ask that the rest of the answer be delivered to Dan in person.